Good afternoon, everybody. I hope you can hear me loud and clear. I'm Dr. Vladislav Kowalski. I'm Chief Scientist at Sanuvox. Many of you know me as the author of the so-called Red Bible. Today we're going to be discussing ultraviolet science and history. And I think we will have a large amount of focus on UV air disinfection, as that is currently a hot topic and offers a potential solution to the coronavirus pandemic, as well as many other diseases and hospital-acquired infections. So, okay, we will get started. I hope everyone can see the screen okay. If not, let me know. This is the first webinar in a series of UV air safety webinars, and we're addressing ultraviolet science and history today. Let's go through the history of ultraviolet disinfection. The history of UV goes way back to 1801, when ultraviolet radiation was first discovered in the spectrum of sunlight. Fraunhofer mapped the spectral bands, and Wheatstone, famous Dr. Wheatstone, produced the first mercury vapor lamp for research purposes. Stokes, the famous Mr. Stokes, developed the quartz lamp for experimental applications. In 1877, the bacterial effects of UV were discovered, and that began the history of UV disinfection. The first experiment on bacteria was performed by Geisler in 1892, who demonstrated that these new arc lamps were lethal to salmonella. The spectrum was completely mapped out in 1903. The first quartz lamp for applications was developed in 1904, and UV was first used to disinfect water in 1906. And the first large-scale application for water disinfection was in 1909. The water industry has had over 100 years of successful application of UV disinfection. But from here on out, we're going to be looking at UV air disinfection and surface disinfection. In 1925, the first attempt to use UV for air disinfection was reportedly done by Regard, although it may have been done earlier in the 1920s. We don't know the exact year. The first overhead UV system was used in hospitals to disinfect the surfaces of patients' wounds, as well as disinfect the air. The first application of upper room systems for disinfecting air was 1937, thanks to Dr. Wells. Rentschler applied systems to an air conditioning system to disinfect air. And it was shortly thereafter UV in the form of upper and lower room systems were applied in 1942 in a military barracks. The first placement of a UV system on an air conditioner, not only for cleaning the air, but for disinfecting of mold, occurred in 1954. It wasn't until 1957 that Riley demonstrated without question that UV could eradicate TB bacilli in the air, and it was some 30 years later the CDC acknowledged that UV could eliminate TB. The WHO followed suit in 1999, and the first actual study in a human building by Menzies showed that induct UV reduced respiratory systems. This brings us to the present. In 2022, EPA, among other agencies, recommended the UV air cleaning in the wake of the coronavirus pandemic. UV occupies a range of the electromagnetic spectrum. You can see the rainbow segment here represents visible light. Just below that, we have UV rays. We have UVA, UVB, UVC, and vacuum UV. Of most interest to us is the UVC range from 200 to 280 nanometers. In particular, the spectrum of 254 nanometers is the most commonly used spectrum in UV air and surface disinfection and water disinfection. And there's the growing interest in far UV, which extends from 200 to 230 nanometers. Oops. Didn't mean to jump ahead there. Now, ozone production occurs about 185 nanometers and above. Vacuum UV below 200 nanometers does not penetrate air very well. And in fact, it almost immediately forms ozone and hydroxyl radicals. So the ozone literally seems to come right off the glass. UVB, UVA are of less interest to us, as is near UV at 405 nanometers, and that is a range that we also call blue light. Ultraviolet science. Ultraviolet light in the UVC range, particularly 254 nanometers, is considered germicidal, or as we say today, antimicrobial. Photons are absorbed by the base pairs of the DNA double helix or the RNA, if it happens to be an RNA virus. This disruption of the DNA inhibits DNA replication and function. Photodimers are induced at pyrimidine bases, T's and C's, thymines and cytosines. The purines, adenines and guanines, also form dimers, but to a much lower rate, to a much lesser degree than do the pyrimidine bases, T and C. Now, ultraviolet light, as I have noted earlier, is part of the electromagnetic spectrum. Now, you can model ultraviolet light as photons, or you can model it as electromagnetic waves. Now, in terms of electromagnetic waves, UV light consists of two mutually perpendicular waves traveling along an axis in the direction of propagation. These two waves are composed of an electric field and a perpendicular magnetic field. UV has a wavelength that, of course, is at 250 nanometers. And the direction of the rays shows you that UV has directionality. Energy is absorbed in discrete quanta. And we have learned from Dr. Schreier's research that the exact orientation of the bases makes a big difference in whether or not a UV photon is absorbed. That is, the DNA molecule bases themselves are facing a certain direction. And let's address that, as this has not been well addressed in the literature previously. Ultraviolet electromagnetic interaction with DNA bases. This occurs primarily for thymine and cytosine molecules. Now, both of these pyrimidines have what are called a dipole moment, an electromagnetic dipole moment. Each molecule has a positive end and a negative end, and somewhere in between there is a point at which the dipole moment can be identified. The absorption of quanta of energy will occur when the dipole moments of the bases are suitably oriented with respect to the incoming electromagnetic waves. For thymine, the dipole moment has been identified at 4.34 D, I believe it stands for diameters. And if the UV ray is coming in perfectly aligned with that dipole moment, there's a high probability the photon will be absorbed. Cytosine has a much higher dipole moment, but still it will absorb UV rays, UV photons, although at a somewhat lesser rate. Energy will jump from the ultraviolet waves to the molecule, creating an excited electron state. The excited bases will break their hydrogen bonds, and fuse with adjacent pyrimidines. The directionality of the UV rays when they are absorbed by the pyrimidine bases accounts for the higher inactivation rates in air versus surfaces, and I will demonstrate that in a later slide. Purines, as I've noted, are much less reactive than pyrimidines, so we won't address those directly here. Ultraviolet photodimerization occurs when two adjacent bases fuse to form a dimer. These dimers inhibit replication and, in effect, kill or inactivate the virus or the bacteria or the fungi. Thymines preferentially absorb ultraviolet photons, and they bond with adjacent bases after severing their hydrogen bonds with adenine on the complement DNA strand here. Here we see a T to A with two hydrogen bonds. It absorbs the UV photon. The hydrogen bonds break and the thymine bonds with the next nearest neighbor, or usually an adjacent thymine or cytosine. Oops. The blue are the hydrogen bonds. The red are covalent bonds between the two thymine molecules. The probability of cytosine dimerization is always a bit lower than for thymine. Besides formation of dimers, DNA crosslinks may be formed, as well as links between DNA and proteins. But crosslinks can form between the template and the complementary strands of DNA, and they can form between either strand and protein molecules. The effect of DNA crosslinking may not be very significant with 254 nanometer radiation, as this primarily forms pyrimidine dimers, and those are sufficient to inactivate a virus or kill a bacteria. There is some suggestion in the literature that far UV produces less dimers and may be producing more crosslinks, either to proteins or between strands. Future research should elucidate that mechanism for us. Photoreactivation is a factor you might be familiar with. UV light can both create a dimer and it can break a dimer and reform a bond. The process happens again and again while DNA is being exposed to UV. Any given base may go into and out of a dimerization reaction with adjacent neighbors, or it may go back into a bond with its hydrogen bonded complement. At some point, the bond is sealed and the dimer is formed, or the dimer will not be formed if the bond photoreactivates, which can also occur from visible light. Now, photoreactivation requires enzymes to reconnect the dimers. And since very few viruses have the ability to use enzymes, we don't see a lot of photoreactivation in viruses. We see it mainly occurring in bacteria. In some cases, we know the degree to which photoreactivation occurs, but in most cases, we don't know the exact rate of photodimerization. However, we do assume that any photoreactivation that occurs as part of the disinfection process. Now, it could be noted that medium pressure UV lamps, which have a broad band of UV radiation, can inhibit enzymes, and therefore there may be less photoreactivation when we use medium pressure lamps as opposed to using low pressure lamps. Let's look at the ultraviolet lamp spectra of the common lamps. Low pressure ultraviolet lamps have a peak around 254 nanometers, and this is the red peak we're seeing right here. This is a low pressure lamp. Medium pressure UV lamps have a broad band of UV radiation from about 250 to 320. LEDs can have very discrete spectra, such as at 270 or 265 or any other frequency that we are interested in. Far UV lamps have a peak around 220 nanometers. Here's an example of a far UV spectrum. Now, each of these spectra can have a different germicidal effectiveness. In the example I'm showing here, we're looking at the germicidal effectiveness on the bacteria E. coli. Low pressure UV lamps sit at about the 85% effectiveness, germicidal effectiveness of E. coli. Medium pressure lamps may not sit at that point on the germicidal effectiveness curve, but they sit at multiple points. And the cumulative effect of all these multiple frequencies on the kill rate makes medium pressure lamps operate very similarly to low pressure lamps, except that medium pressure lamps tend to produce ozone. LEDs have a single peak spectra, and that can occur anywhere. It can be right at the germicidal effectiveness peak, about 265, 270. And studies have shown that indeed 270, 265 nanometers produce germicidal effectiveness very comparable, if not greater, to that of low pressure lamps. The far UV spectrum is from about 200 to 230. I don't believe that we actually know the germicidal effectiveness of E. coli below this range, since this was from an old study, but we will see in the following diagrams that it extends upwards. UV LEDs can be very effective at right frequencies, but they're also considerably more expensive and more costly than low pressure UV lamps. Now, instead of looking at E. coli for our germicidal effectiveness, we can take a look at the spectral response of individual DNA bases. Thymine is the primary target. You see it here in the red line. It's the primary target for absorption of UV photons. Cytosine, the other pyrimidine, is the secondary target. And you can see cytosine here in the blue. Now, there's a curious fact that the cytosine graph and the thymine graph intersect at approximately 254, which suggests that they would have approximately the same rate of UV absorption. And that may be true. However, the bond energies for cytosine are higher. Therefore, we get less absorption. Even if the frequency is good, we still get less absorption by cytosine. We see the same effect for adenine and guanine. We can see that adenine and guanine actually have higher absorption rates than thymine or cytosine. And yet we see very little dimerization of adenine and guanine. That's because the bond energies are so high that we do not see a lot of adenines or guanines dimerizing. Some of them do, but mostly it's primarily thymines and cytosines. We can also see that at 222 far UV, the absorption is potentially higher for far UV than it is for UV at 254. This might suggest that far UV would perform a little bit better, and some of the data does indeed indicate that far UV against some bacteria and some viruses may perform better. But they do really, seriously, the far UV and 254 nanometers produce approximately the same average kill rates within some range of uncertainty, the error of testing. So they operate very similarly, about the same level of performance. Now, relative humidity can affect the performance of UV in air. That's because we have two forms of DNA, aDNA and bDNA. aDNA is dry or dehydrated and shrinks. It shrinks like in the same fashion that horsehair shrinks on a relative humidity indicator, for the engineers that are familiar with that. Under dry conditions, the DNA will shrink and it will compress the bases, bring them closer together. Whenever the bases are brought closer together, the probability of dimerization is increased. This was shown, I think, in the Schreier experiment, among others. A high relative humidity, when bDNA is formed from the absorption of water molecules, the DNA stretches out and the bases are more separate. As a result, it becomes more resistant and has a higher sensitivity or a lower sensitivity to UV. So let's compare air, water, and surface UV disinfection. We can see on the table on the right, which is an average for D90 values of viruses. For viruses in air, they don't vary much. Bacteria do, but not viruses. We get approximately the same kill rates at high relative humidity and low relative humidity. In water, we get D90s that are much higher, easily 10 times higher than air. And on surfaces, for this set of data, the surface resistance to UV is somewhat higher than water, although this varies, the surface resistance and the water resistance of microbes looks very similar on the average. Airborne microbes are subject to diffusion and turbulence. This means that they are spinning and they are in motion. As a result, they are exposed to ultraviolet rays from all sides. Surface-borne microbes do not have any motion, presumably are not in motion, and so the exposure is limited by the direction the UV light comes from. And I'll show you in a moment why there's a difference there. Waterborne microbes are assumed to be in dynamic motion. However, water attenuates UV, and as a result, the UV susceptibility in water is very similar to the susceptibility on surfaces. Now, there are three types of ultraviolet light delivery modes. There is the collimated beam or parallel ray mode, which we have seen many times used in experiments used to measure the UV dose of microbes. Collimated beams are parallel rays, similar to what you might see in a laser. Now, a point source, which is very well approximated by LED sources, and LED acts very much like a point source, causes the rays to expand outwards spherically in all directions. The third form of UV light that we know of is we call multi-vector light. It could also be called diffuse light. Diffuse light is like the light from clouds that comes from all directions. But for UV, we call it multi-vector light because the light converges from multiple directions. When you have multiple lamps in an enclosure with reflective surfaces, you will end up with multi-vector light. Now, multi-vector light tends to be faster and more effective than, say, a collimated beam or a point source due to the fact that it gives more all-around exposure to the microbes. This is very similar to when a microbe is spinning in air. You could say that a microbe floating in air in dynamic motion is experiencing multi-vector light exposure, even if it was in a collimated beam field. Now, the ultraviolet inactivation of microorganisms is generally observed to be described by a two-stage process. There is a first stage, which may go on for 2, 3, maybe 4, even 5 logs, but at some point, the slope of the survival curve alters dramatically and makes it difficult to achieve further log reductions. The microbes that are in this group, we call it the second stage, act like they are resistant. But in general, this resistance is an artifact of the fact that these cells get clumped. They clump or cluster together or aggregate, and the cells inside the aggregated clumps are protected from UV, so they behave as if they are more resistant. There really is no second population. This is an old idea that there are two populations. It's just that some of the microbes have protection from UV, and so they appear to be more resistant, although their intrinsic resistance is no difference. Survival tails are generally an artifact of the preparation process. If the preparation process enhances clumping and clustering, you will get a survival tail. If there are impurities in your solution and the microbes are protected, you will get a survival tail. In theory, the first stage rate constant is a single stage and an intrinsic property of the RNA or DNA, and will completely define the response of the microbe. Now, I've mentioned cluster formation. UV is scattered by particles in the 0.25 to 2 micron range because of the fact that they have harmonics, which literally harmonize, resonate with the frequency of UV and the size of the microbe. The DNA core of a microbe that's about 2 microns might be 0.25 microns. And as you can see, 0.25 microns is identical to the wavelength, 254 nanometers. This causes resonance and harmonic effects. You can see these harmonic effects when we plot these effects in terms of the shielding acquired from scattering. There is a major inflection point at 254, also at double 254, at triple, and at quadruple. We also see it at 8 times 254, an inflection point. Clumping and photoprotection can provide photoprotection to cells when they are stacked or when they are in clumps. If you have one cell downstream of another cell, and this cell blocks UV or scatters UV rays, the second cell will see reduced UV irradiance. If there is a third cell, the process will continue. And if you have 10 cells in a row blocking each other, that 10th cell will have a great deal of photoprotection. Clumping occurs when we put samples on slides or on Petri dishes and allow the sample to evaporate. On the left here, we're looking at clumped C. difficile spores. These spores were inoculated on a steel disc, and when they dried, they formed major clumps all around the meniscus of the drop. Now, we sprayed Staphylococcus aureus cells on glass, and we got no clumping at all. For the C. difficile cells, we saw a major survival tail when we irradiated. But when we irradiated Staph aureus, we got essentially a direct path to 6 logs of reduction with no survival tail. These are some examples of survival tails. Spores also form shoulders because they can resist UV to a degree, but most microbes tested in the laboratory will form tails, survival tails, as a result of clumping, as a result of the preparation procedures. Ultraviolet genomics is one of my favorite topics. I'm showing you here the photodimerization probability of a sequence of bases, including G's, A's, C's, and T's. The photodimerization process is considered sequence dependent because thymine dimers outnumber thymine to cytosine dimers, which outnumber cytosine to thymine dimers, which outnumber cytosine to cytosine dimers. And all of these types of dimers outnumber NA-purine dimers. This is a set of actual data from Becker and Wang for sea urchin DNA that they irradiated. We can see that the thymines have the most reaction and go to a maximum after about 10 bases. The cytosines also have a maximum when they are not affected by the surroundings. We can see that this cluster of pyrimidines is self-amplifying itself to form what we call a hotspot. These genomic hotspots exist everywhere. This is an example of all the hotspots in influenza DNA virus from the first base to about the 4,000th base. There are a great many dimers, potential dimers or potential hotspots in any genome. For example, here's Borrelia afzelii. Again, we look at the first 4,000 base pairs and we see a lot of hotspots. The red hotspots are pyrimidine hotspots. The yellows are where purines occur in the hotspots. These hotspots are very common in any genomes and in fact can consume the majority of bases. Here's a poster of ultraviolet D90 doses for viruses. We have measured doses, D90 doses for many viruses, but for the ones that we do not know, I have used genomic analysis to compute them. And you can see in this this chart of ultraviolet D90 doses that if we look at 40 millijoules per square centimeter, we're effectively going to take care of well over 90, perhaps 95% of viruses will be encompassed. In fact, if we use 5 millijoules per square centimeter, which is 50 joules per square meter, we can cover about half the viruses, including all the major cold viruses, influenza, and SARS coronavirus, which sits down here in the lower right-hand corner, being the weakest virus of all. In fact, it actually sits below the curve of 1 millijoule per square centimeter. I had to double its D90 just to get it to show up on the chart. Here's another chart of bacteria. In bacteria, most of the bacteria tend to cluster below a D90 dose of 10 millijoules per square centimeter. That means a dose of 10 millijoules per square centimeter will effectively eradicate most of your viruses. The ones that cannot eradicate easily are often the spores. But as I will show later, it makes more sense to remove spores with a filter than with UV. Both of these previous posters are available from Sanuvox on request. Now let's look at types of air disinfection systems. I'm going to run through this fairly quickly. There are basically 4 types: in-duct UV, portable UV, upper room UV, and whole room UV. Now, in-duct UV and portable UV units are seen to be virtually identical. It doesn't really matter if the unit is outside or inside the room. Each unit will have an airflow, an efficiency, and when these units are applied to a room with a volume V, we can compute the air change rate, the unfiltered or uncleaned air change rate. We can also compute the equivalent air change rate by multiplying the efficiency times the airflow divided by the volume. This gives us our equivalent air change rate. Now, an upper room system has an upper room zone, which is much like a duct that is missing one wall and through which the air both enters and exits. In this example, we're showing ceiling fans because upper room systems are often augmented with ceiling fans. Again, the upper room has an airflow and efficiency through the zone, and in any given room of volume V, it has an air change rate, and the equivalent air exchanges can be computed by multiplying the efficiency times the air change rate. Now, the whole room UV system can be used to disinfect air, although they are generally used for surfaces. The whole room system is like an inversion of the in-duct system because the UV lamps are in the room. However, the fan produces, if there is a fan in the duct, it produces an airflow. The UV system, the room UV system as a whole, has an efficiency for a room of volume V. The equivalent air change rate is simply, again, the efficiency times the air change rate. There are many UV air disinfection standards, also for filters, electronic air cleaners. For all these standards, we measure disinfection efficiency as a performance parameter and airflow. There are 2 types of tests. Test 1 is an in-duct system, or for an in-duct system or portable air cleaner, merely measures the efficiency across the component and ignores what it does to the room. The test type 2 is modeling in a model room at steady state conditions, with the air cleaner either on or off, and the ratio between those conditions gives us our efficiency, our disinfection efficiency. These types of tests can be performed for filters. There is only one test from ISO which looks at UV dose, but in general it is not necessary to test the UV dose or the UV irradiance, as long as you can measure the disinfection efficiency and measure the airflow, you know everything you need to know about that system. The steady state system test puts a source S of some contaminant into a room and operates the room without UV until a steady state condition is reached with a high level of contamination. Now, when they turn the UV system on, that high level of contamination will drop way down. The difference between those contamination levels is implicitly the disinfection efficiency of the system. It doesn't matter if it's an upper room, an in-duct, a portable, or a whole room system. This test measures the disinfection efficiency the same for all 4 systems. That is, it puts all 4 systems on a comparable basis. Airborne concentrations, when we put a constant source into a room, will cause the concentration, say in CFU per cubic meter, to increase over time. In this case, we're looking at outside air at 15% of 2-ACH, which is about 0.35 ACH. The outside air does not really reach steady state after 8 hours. The level of contaminants continues to increase. Now we compare it with an upper room system in the red, which includes that 15% outside air, and it reduces the concentration to less than half of what it was. If we use a filter, a MERV13 removing 98% of the particular microbe that we're testing here, we get a major reduction in the green. And if we use ultraviolet light with a 99.9% reduction, and this is from an actual test, all these values are from an actual test, we see the largest reduction in concentrations occur from the induct UV system. Now let's compare induct, outside air, and upper room systems more directly. This is a logarithmic curve, so there's straight lines. The outside air takes 8 hours to reduce the indoor concentrations by about 1 log, and that's at the typical 15% of outside air. The upper room system reduces it just over 2.5 logs in 8 hours. But the induct UV system, the most efficient of all these systems, reduces it by 6 logs in less than 6 hours. Induct systems will typically outperform upper room systems. The reason is upper room systems have low UV power, they have low airflow, so you can't expect their performance to approach that of an induct system, which typically has high airflow and high UV power. Now, theoretically, if you boosted the power and airflow of an upper room system, you would approach the performance of an induct UV system eventually, but you could never exceed the performance. The induct system represents the theoretical maximum of performance for any given airflow. Now, one issue I feel important it is to cover is the combination of filters and UV. This is an ideal combination because the filters remove large spores and bacteria, as I mentioned before, whereas the UV is capable of removing all of the smaller bacteria and viruses. It's easy to compute the total efficiency of a combined removal. And currently, MERV13 filters are being recommended by ASHRAE. We combine a MERV13 with any UV system, and you will remove microbes across a large area. You remove a large area of microbes. There's the end of our introductory session on ultraviolet science. I've finished a bit late, but we still have plenty of time for questions. So if there are any questions, I think I can take them now.
Yes, if you could type in questions or raise your hand and we can deactivate your mic, that would work fine.
Okay, I'm going to unshare lead.
I think Dr. Kowalski inadvertently left the meeting.
Okay, I'm back. Can everybody hear me?
Yep.
I'll gladly take any questions if anybody has any. I think, Paul, I'll let you coordinate.
Yep. There are some people, Dr. Kowalski, that asked if they could get the slide deck following the meeting. Is that all right with you?
That's fine if it's okay with you. I'll let you take care of that, Paul.
Perfect. Well, it was a very informative session, that's for sure. Better understanding on how the impact of UV radiation has on different types of viruses, and especially the graphic of the size of the different viruses that can be impacted by the light was I think an informative piece of information.
Okay, Paul, were there any typed in questions for me to address? Because I don't see them here.
No, there wasn't any questions specifically. So I guess
perhaps we can open it up to a discussion. People can turn on their mics and ask questions directly or make comments if they would like to. Does anybody have anything to say or a question to ask? I think you can raise your hand or just turn on your mic and start speaking.
I'm trying to see if I can reactivate everybody's mics.
Okay, if not, if there are no questions, I suppose we can end the session here and now. I'd like to thank everybody for joining. I hope you found it informative and interesting, and I hope you will join us on the next webinar 1 month from today. Thank you very much. Oh, here's a question.
Yeah, they're starting to come in. Hold on a second. Let me just.
Sam Guzman asked me if the cumulative effects of medium pressure lamps equal that of the low pressure lamps. What wavelengths do you think are responsible and what is the mechanism? Well, seriously, all the wavelengths summed together or integrated, as we say, would provide the same dose as the single spike at 254. And since the dose is approximately equivalent, we get approximately the same effect from LP and MP lamps at approximately the same wattage. I hope that's clear. Okay. If there are no further questions, I don't see any other than the comments. Thank you for all the wonderful comments. We will get you the slides. And Paul will be able to send you the slides, the recording, or whatever when you want it. Thank you very much. I hope you found this important, interesting, and informative.